Okay, so my name is Jake Jaffe, and I'm going to tell you about my final project, which is a web application called peptide2genome. Just some quick highlights here. Peptide2genome is really meant to be a useful bioinformatics tool for protein biochemists like myself who find themselves living in now the genomic era. But as far as this class is concerned, the highlights are it's a Django-backed web application. It uses external data through an external RESTful API to gather information to be entered into this database application. And it uses something what I would kind of describe as database memoization to make popular queries extremely fast. It tries to remember what's been done before and benefit from that. It also uses a smart hashing system for database ID entries to avoid ever repeating work or creating entries unnecessarily. And then it tracks popularity of queries, making potential future aggregations or interesting data mining possible. But I just want to take a very quick moment to explain the biology here, which sort of underpins the reason for creating this application. I'm sure everyone here is aware that the human genome consists of DNA. DNA is a very long string of nucleic acids. The alphabet of the genome is basically 4 different letters. And most of the DNA in the genome doesn't really do anything, but every once in a while we encounter these things called genes. And so this might be an unimportant part of the genome here and here. But in the gene, there are these funny kind of structures, and they're divided roughly into 2 camps, something that's called an exon and something that's called an intron, which we'll talk about in a second. But when genes get made or copied, they are first transcribed, and they're transcribed into RNA, and they get copied with both the intron and the exon parts present. Then a process called splicing happens, where these introns are cut out and the exons are pasted together end to end. This goes on to be translated into proteins. Now, instead of having 4 letters of nucleic acids, proteins have a 20-letter alphabet of amino acids, and it takes 3 bases of DNA or RNA to encode 1 letter of a protein, but it's also a degenerate code. So in my day job, what I do is I take these proteins and I chop them up into even smaller bits, and I read out the sequences of these smaller bits. So here's the main problem I'm trying to solve. Given one of these smaller bits or a short piece of protein, where might it be found in the genome? And there's several important scenarios to consider. One is that there may be multiple copies of a gene in the genome, and that's illustrated here, where maybe each tick mark indicates a place that a gene or a short segment of a gene might be located across the 23 chromosomes in the genome. Or maybe you have some bit of a protein that spans more than 1 exon and spans this intronic region that gets sliced out. Now, this kind of positional information is really the currency of modern genomics, but it literally is being lost in translation here. And so I wanted to develop an app to map these things back. Some characteristics of the app that I was thinking about, I wanted to ultimately be very fast, be able to crunch tens of thousands of these short peptide sequences and return the positional information extremely rapidly. I wanted to be smart and not repeat work that had been done previously. And I wanted to be a little bit sneaky, which is to keep track of what I or other people in my group might be searching for mining later, trying to see what are the popular hotspots that people keep coming across. Okay, so I decided to develop this application using the Django framework, and there are 4 really kind of basic model tables that are part of it. One talks about proteins, these long sequences of amino acids, and just basically holds some accession number that I can refer to this with this external REST API that I'm using to gather positional information, and then the sequence of it. And then we have peptides. These are the small bits that I care about. This has also a sequence, and then this is where I start to keep track of how many requests are made for a given short peptide sequence. And then to deal with this notion of these cutting and pasting events that happen in biology of exons and introns, I have this idea of a span group, which talks about the main area that a sequence is coming from in the genome, and then regions, which are the specific bits that are cut and pasted. Then I have a couple of special bridging models that, that help relate either peptides to these span groups or span groups to the very specific regions. And you'll see here that these consist totally of foreign keys to those more basic models, and that the ID, we'll talk about in a second, is just going to be some unique hash value. So this structure allows for possible many-to-many associations. Django does support many-to-many associations in basic models, but it doesn't have great support for constructing primary keys that are composites of foreign keys. So that's why I decided to go with this more classic database architecture here. And this is now a pseudocode representation of the main logic loop of the program, but a request is submitted for a peptide, and if it can find that peptide in the peptide table, it will simply spit out the locational information about that. But if it can't find it, it will attempt to first make sure that it's in the protein database, and if it is, it will go out to the web and do the very careful mapping, and then incorporate those results into the model and ultimately give it back just as if it had been there. And if it's not really in any protein anywhere, it will give back an error message. So as I mentioned, it uses this Ensemble REST API to get location information. And as a result of this, the first time that you look up the positional information about any given peptide, it's slow, but then the information is cached into these models, and that's why I sort of call it database memoization. Some other features is that all the ID fields in these tables are actually based on hashes of the content. And then the bridging table ID fields are hashes of hashes. Just to give you an idea of this, so representation of a span group here, this primary key is a hash, and then this hash is formulated based on a catenation of all these data, so it's guaranteed unique, but deterministic. So if I ever get this region again, I simply have to check to see if it's in the database by calculating its hash. And then these bridging tables have the same sort of property where their main entries, whether they're these foreign keys or the primary keys, are all hashes. So in this case, this span group ID is referring back to this one, and this one is referring to this one. And then this hash is calculated based on the catenation of these data. So this allows me to handle responses and not repeat work. If I have gone out and determined some locational information from afar, I simply need to handle the response table I'm getting from the external API, and then check to see if this deterministic hash that describes the information within it is present already in any of my models. And if it is, then I can just continue on to the next step of what I'm doing. I don't need to create any new objects. And the main idea here is to never create anything that already exists and guarantee uniqueness in the databases. Okay, so that kind of describes the application, but now let's do the thing that's always real difficult for software engineers, and which is the live demo. And so I've got a couple of things to try. Stuff that may or may not I may or may not have searched for before, but here, let's say I want to take some arbitrary piece of this protein, which I'm selecting on the fly, and now here's really the front end of the application. Very simple, kind of Google Omnibox-like interface. I'll just paste that sequence in here. Now, the first time I look for it, it might take a little while. And here, what I can see I'm getting out of this is Here is the sequence that I input. This has only ever been requested once, which was just now when I did it. And now I can actually find that it's in 2 different locations in the genome. So there might be 2 copies of the gene to which this belongs. So now if I want to go back and look for this one again, it's going to be extremely rapid. Now my requests have incremented. The genomic locations here are still the same as before, but we were able to return the data much more quickly. Now let's try some other thing. I'll try this sequence from some other protein called collagen, and I'll take a really long piece of it here. Let's go back and try, and we'll just cut and paste this in here. And it didn't take too long. Here, we can see again, it's the first time I've requested it, but now this bit is only in one location in the genome. However, it has 4 different segments. That means there are a few of those cutting and pasting events that you may have, that I referred to earlier. You know, the future extensions of this will be to actually build in links to see the detailed information about what these segments are, really to set up an API to return these data, as well as sort of feeding into other applications that rely on these genomic locations. I did expose the basic API for all the tables, but moving forward, I want to build out the API to spit back this information, not for just single queries, but for long lists of queries. And then just to show you, it will complain if you can't actually find the peptide anywhere in the proteome. So that's about it. It was a lot of fun to generate. I'm a little bit surprised that I was able to pull it off so quickly, but I guess that's a testament to what I've learned in the class. And thanks for listening.